Hi everyone, welcome back to my channel. As a continuation part of meiosis concept, in this video I'm going to talk about second meiotic division or meiosis II. Meiosis II begins as soon as a meiosis I is over or soon after the completion of telophase and cytokinesis, meiosis II begins. So there is no process of DNA replication or DNA duplication after the cytokinesis of meiosis I. Soon after cytokinesis, meiosis II begins. This meiosis II is of simple process and it's of short duration when compared to meiosis I. As you all know, meiosis I is of long duration and it's a very complex process. Whereas meiosis II is a very simple process and it is more or less similar to that of mitosis. As a result of meiosis I, it produced two haploid daughter cells. As you can see in this diagram, these are the two haploid daughter cells which are produced as a result of meiosis I. And these two haploid daughter cells now undergo second meiotic division or meiosis II. As I already told, meiosis II is more or less similar to that of mitosis. Even meiosis II has mainly four substages like that of mitosis. They are Prophase II, Metaphase II, Anaphase II and Telophase II. So these are the stages of karyokinesis. Karyokinesis means division of nucleus. So we'll discuss these four stages one by one. First one is Prophase II. So this is the Prophase II step which is similar to that of mitosis. Even in the Prophase II, the condensation of the chromosome begins. The chromosome was long, thin thread-like as a result of decondensation or uncoiling of the chromosome in the telophase and cytokinesis of meiosis I. As a result of that, the chromosome was long, thin thread-like. This long, thin thread-like now start to coil or become folded, and that process of coiling of DNA is called as condensation. So condensation begins in the Prophase II stage. Apart from that, the nuclear envelope which is present around the chromosome now start to break down and it finally disappears. The nucleolus also become disintegrated and finally disappears. The spindle fibers are formed between the two centrioles and these centrioles move away from each other, and by the end of this Prophase II, the centrioles reaches the two opposite poles of the cell. So that is about the Prophase II. Next stage is Metaphase II. In the Metaphase II, the spindle fiber which are formed between the two centrioles now get attached to the kinetochore of the centromere. Kinetochore is a disc-shaped structure which is present on the surface of centromere. So this spindle fiber get attached to the kinetochore of the centromere of each chromosome, and they bring this chromosome to the center of the cell. As you can see in this diagram here, the chromosomes are aligned or they are present at the equatorial region or to the center of the cell, or they are aligned at the metaphase plate of each cell. So that is regarding the Metaphase II. Next stage is Anaphase II. In the Anaphase II, the splitting of the centromere takes place, or the centromere now separate or splits, due to which the sister chromatids of each chromosome will separate because of breaking of the centromere. And these separated chromosomes or chromatids now move away from each other and they start to move towards a different pole. As you can see in this diagram, this is a one chromatid moving towards the north pole and this is another chromatid moving towards the south pole. So two separated chromatids move towards the two opposite pole of the cell. During the movement of this chromatid, the kinetochore is towards the centriole and the arms trails behind it. So that is about the Anaphase II. Next stage is Telophase II. In the telophase II, the sister chromatid reaches the two opposite pole of the cell, and after that this chromatid now undergo decondensation or uncoiling to become a long thin thread like a chromosome. Apart from that, the nuclear envelope is also reformed around this decondensing chromosome. So these are the chromosome and nuclear envelope is reformed or formed again around this chromosome. The nucleolus is also reformed. The spindle fiber which was present between the two centriole now break down to form a tubulin protein. So these are the changes that takes place in the four stages of the karyokinesis. After the karyokinesis, cytokinesis takes place. Cytokinesis means division of a cytoplasm or division and distribution of cytoplasm to the produced daughter cell. So by the end of cytokinesis, 4 haploid daughter cells are produced, having haploid number of chromosome and equal amount of cytoplasm from the parent cell. So in the meiosis, single diploid cell produces 4 haploid cell. So diploid condition is changed to haploid condition, and this meiosis process takes place mainly in the gametogenesis, which results in the production of a haploid gamete. So that is all about the process of meiosis in detail. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.